Okay guys, so let's start this video by going over what we ended on the last video. So I didn't quite get to cover the action in muscular contraction last time. So let's go over it again, maybe a little bit slower. So remember, when we're talking about how a muscle contracts, first we're talking about how it receives the message. Why does it need to contract? And since it's skeletal muscle, we decide that. So it comes from our brain. And in order to come from the brain, it needs to come from the nervous system. So we're going to need a nerve cell, which is called a neuron. Now, the neuron doesn't connect directly to the muscle tissue. It'll come really close to the muscle tissue, and it'll come close to a specialized place on the muscle tissue that we call the motor end plate. Okay, so we have our neuron, which in this case is going to be a motor neuron because it's going to cause movement, and then we have our motor end plate, which is going to be the place where the muscle receives the signal. And then, since it's a synapse, so it's a gap, we're going to need something that can communicate the message from the neuron to the muscle, even though there's a small gap there. And what does that is a neurotransmitter. Now, there's several different types of neurotransmitter in the body, but for muscle contraction, the main one we're going to talk about is acetylcholine. Okay, so the motor neuron brings the signal down to the synapse. The synapse has has vesicles there full of neurotransmitter, in this case acetylcholine, and the acetylcholine is released into the synapse, so the gap between the neuron and the motor end plate. The motor end plate is the specialized part on the muscle fiber that has receptors for the acetylcholine. So the acetylcholine takes the message from the neuron across the synapse into the muscle, and that's when we get contraction of the muscle. So here is another little bit of information about how it goes. The neuromuscular junction will allow the motor neuron to stimulate the muscle fiber to become electrically excited. Now, electrically excited just means the transfer of electrons. Okay, that's what electricity is. And all of the communication between our brains and the rest of our body through our nervous system is basically electrical signals. Moving electrons around on the muscle membrane, and that passes signals through our body. Now, movement of these these electrical signals is called an action potential. Okay, and we'll talk a lot more about that when we do the nervous system. The action potential is what stimulates the muscles to contract. So when we get down to the neuromuscular junction, the neuron has carried this electrical message all the way down from a relay center, probably in the spinal cord or maybe even in the brain, and it gets down to the synapse. When we get to the synapse, then that acetylcholine is going to be released from the motor neuron into the synaptic cleft. Now, if you see that word synaptic cleft, that just means the empty space around the synapse. Okay, so the synapse and the synaptic cleft kind of means a similar thing. So the acetylcholine will diffuse across the synaptic cleft towards the motor end plate. Okay, so that's the part on the muscle fiber. When the acetylcholine binds the receptor on the motor end plate, that will stimulate the action potential into the muscle fiber. That'll cause the muscle fiber to contract. And if we go back to our little picture here, we can see all of that happening. Okay, so our neuron will bring the electrical impulse, the action potential, down to the synapse, and then when we get to the synapse, we'll have those vesicles that are filled with acetylcholine. They will move, so that acetylcholine moves across the synapse and attach to a receptor for the muscle fiber. Once that happens, then the message has been carried from the neuron through the synapse and into the muscle. So if we go back to that first picture there, B, if I erase that arrow and I draw a new one, the action potential will travel down here. It would have traveled through the acetylcholine and then moved into the muscle, so that the muscle gets the message from the central nervous system. Alright, so let's talk about our first big physiological process, and that's going to be muscle contraction. Okay, so when we talk about contraction in muscles, what we're talking about is the ability or the capacity of a muscle fiber to undergo shortening, to change their shape and to become thicker. So contraction is when muscle fibers get shorter, and when they do that, they get thicker. In order for that to happen, there's two proteins that have a major role to play. These are actin and myosin, or myosin. Now the actin are going to be thin protein filaments, and they're going to be kind of lightish in color. The myosin is going to be thick protein filaments, and they're going to be quite dark in color. Now if you think back to the appearance of the skeletal muscle, we get that striped appearance where, you know, it looks like little stripes are going across the muscle fiber. Well, it's these alternation of these actin and myosin fibers, light and dark, light and dark, light and dark, that gives that muscle its striated appearance. Okay, so that's what gives the striations, is these filaments of actin and myosin. Now these filaments will overlap, just like if you were to cross your hands and your fingers slide in between each other. That's going to be exactly the process that happens when myosin and actin overlap. What's going to happen is not only will the fingers, your fingers and the muscle will get shorter, they'll also get thicker, and that's exactly what contraction is going to be. Now, the subunits that contract, so the little striations, if you want to think about it that way, those are going to be called sarcomeres. And a sarcomere is a band of myosin and actin filaments on either side. And I'll show you a picture of that in a couple of seconds. So here's a look at an electron microscope, an electron micrograph of the muscle fiber, muscle tissue. So you'll see the myosin fibers right here, and then you'll see the actin fibers right here. So the myosin fibers are the thick, dark ones, and then the actin fibers are the short, thin ones. And then when you look at it down here in terms of a picture, a cartoon, then you can see the actin filaments right there, and then the myosin filaments are the thick red ones along here. So this is going to make up a single sarcomere. So this right here is going to be a single sarcomere. Okay, let's keep that sarcomere in mind. And now the sarcomeres are going to contract via the sliding filament mechanism. Okay, so think about when you fold your fingers or your hands together and your fingers slide over each other. Now the way this works is myosin have these extensions that come off the protein line, or the configuration of the protein, and these myosin heads, they'll attach to actin and they'll form cross bridges. Now, using energy that's in the muscle, myosin heads will actually pull the filaments together within the sarcomere. So when it does that, it moves the sarcomere, it moves the actin filament over top of the myosin filament a little tiny bit. And when it does that, then the muscle fiber will actually shorten. And you've got to think this happens over every time you see a striation in a muscle fiber, that contraction is happening. So even though it moves a little tiny bit, it's enough to move the whole fiber a fair bit. Now, once those myosin heads contract and move the actin, the heads are no longer in a position to make any more progress. So what we do then is we use energy in the form of ATP, and we detach those myosin heads from the actin, and the myosin heads go back to their original position. But now those myosin heads are ready to make another connection with actin and then contract and make that muscle contract even more, even shorter. And we'll see this in the next slide. I'll show you a picture. So if we look at this picture here, we've got the myosin filaments are going to be in blue, and the actin filaments are going to be in red. Now you can see the actin are the thinner, lighter filaments, and the myosin is the shorter, thicker ones. But what you want you to look at is these heads. So the heads are little tiny areas right here at the that branch off of the myosin. Now when muscle contraction is initiated, when it starts, the myosin will come in contact with the actin, and the head will form a cross bridge between the actin and the myosin. While that's happening, if you look between these two pictures here, the myosin head moves backwards. So that's what it's done here. And then as it does that, it will pull the actin into, well, in this case in two directions. It'll move the muscle and it'll get shorter, and as well as that, it'll get thicker. When we use that energy, that ATP, what will happen is we'll break that bond between the head and the myosin head and the actin filament, and then we'll go back to this situation right here. And then, of course, if the action potential is still there, the signal is still there to contract, then we'll have that formation of cross bridges again, and the myosin heads will move back and make the muscle go even shorter. So we'll go around and around in a circle between these two different configurations as long as the central nervous system is telling the muscle to contract. Okay, so we just talked about the basic physiology of how muscles contract, looking at the myosin and the actin filaments. There are a lot of other things that go into the physiology of muscle contraction. You know yourself, when you go to the gym to work out, sometimes your muscles feel full of energy, sometimes they feel a bit heavy, so on and so forth. So we know muscles can look, react, and feel differently depending on their condition. We're going to talk about a few things that have an influence on muscle contraction, and the first one we're going to talk about is calcium. Calcium has an important role to play when we're talking about the sarcomeric contraction of the muscle cell. Now, think back when we first get the signal from the nervous system that travels through the neuromuscular junction, and it activates the muscle fiber. Now, when it activates the muscle fiber, the first thing is going to happen is that action potential is going to message the sarcoplasmic reticulum. Now, you've heard of the endoplasmic reticulum. That's a typical membrane we see in cells. Well, in a muscle fiber, we have a sarcoplasmic reticulum, and it's a series of membranes that travel up the entire length of the fiber of the muscle, and its main function is to store calcium. So when we get the action potential reaching the sarcoplasmic reticulum, what that's going to do is release calcium into the cytoplasm. Now, when calcium is present in the cytoplasm, what calcium is going to do is it's going to move two molecules that generally sit on the actin protein. Okay, now these two molecules are troponin and tropomyosin. The calcium is going to shift these two molecules, and when it does, it unveils, or it makes available the binding sites that are on actin so that the myosin heads can bind to them. So if you want to think of troponin and tropomyosin as a cover, then the calcium comes and takes the cover off the actin fiber so that the myosin heads can bind to it. So once those troponin and tropomyosin are moved, now we can have that sliding filament mechanism work. It can be active, and we can get contraction of the muscle. Now, when the muscle contracts, when it relaxes and it's no longer needed to contract, calcium is pumped back into the sarcoplasmic reticulum, and then that'll prevent the myosin heads from binding to actin, and the muscle will relax and it'll elongate again. So let's have a quick look what happens there. So we get two molecules. We get troponin, these little tiny purple molecules here, and tropomyosin is like the cord that almost looks like it's wrapped around the actin filaments. Now, this is what happens when calcium is not present. The myosin heads can't form cross bridges on the actin fibers because these molecules are in the way. When calcium is present, you can see down in the bottom picture down here, the fibers of tropomyosin and troponin, they'll move out of the way, and then these binding sites for the myosin heads, they will be exposed. That way the myosin heads can bind onto the actin, and we can get that movement backwards, and we can get that contraction.